puedo poner la presentación, el primer slide,
Claro.
que quede ahí abierto.
Claro. Oh, y perdón creo que los de YouTube no me escucharon porque tenía bloqueado el micrófono para YouTube. Vamos a poner mi cámara, de hecho. Ambas cámaras. Oh, tengo mi cámara en el piso, por cierto. Listo. Ahora sí estamos. Buenas tardes a todos.
Se ve bien, ¿no?
Sí, se ve muy bien, se ve muy bien. Buenas tardes a todos. Antes de comenzar, estaba comentando, ya sea la gente en YouTube o gente aquí en Zoom, si nos gusta mandar un mensajito, nada más para saber si prefieren que la plática sea en inglés o en español. Patricia nos dice que prefiere hacerlo en inglés para maximizar el alcance de esta plática, pero bueno, solo díganoslo saber. So, good afternoon, everyone. In YouTube, I was just saying that Patricia is willing to give this presentation either in Spanish or English. So just shoot us a message if you have any preference. We are aiming for it to be in English, but yeah, just let us know if you have any concern about that. But in the meantime, we are just gonna wait maybe a couple of more minutes, and then we can get started. So, hola Jasmine, muchas gracias por estar aquí. Ya veo a Jasmine en YouTube, siempre muy puntual. Muchas gracias. Thanks Jasmine for being here. You're always so on time. Dice Jasmine que genial si se puede en español. Adolf, again, I see you here in Zoom land, so if you have any preference, if you want your presentation to be in English or Spanish, just let us know so we can adjust that. And we are going to get started in a couple of minutes more.
Great. I can give a feedback in Spanish if you would like. So
Yep.
I already prepared the presentation in order to share a little bit with our peers in different countries from Europe and also from the US, because I know that most of these species are well known by these fellows. They have dedicated important time in order to understand the life histories and the relevance of the morfoanatomical features found on these species. So I would like to have this interaction with my peers in other countries because they have the experience and they can add additional information than the one I'm going to share today. So it's open to English speakers, and do not hesitate to consult me and interact with me during the talk, okay?
Perfect. So, yeah, I guess we are already, 5 minutes past the hour. So I think we can get started with this. So before actually just giving the...
Igual en español, o sea, lo vamos a tener allí para las personas que también conozcan del tema. Yo sé que a nivel de Latinoamérica tenemos, a nivel de Suramérica, especialistas que han tratado de trabajar mucho estos grupos y que han ayudado a conocerlos, ya que han sido muy poco estudiados en nuestra región. Existen grandes vacíos, dado a que mucha de la literatura está dispersa en otros idiomas. Claro. Y eso nos limita un poco, pero también somos capaces y creo que entre todos podemos unir esfuerzos y avanzar más en lo que refiere a estos grupos que son altamente complejos.
Claro, gracias Patricia. Pues bueno, entonces de hecho ya se ve que está bien encarrerada Patricia por empezar con su plática. Entonces ya son 5, well, sorry, it was in English, right? So we're gonna get started. It looks like Patricia is all ready to go. So it's already 6 minutes past the hour. So yeah, I think we're good to go. People are gonna start trickling in. We already have a few in YouTube already. I'm gonna be tackling the chat both in Zoom and YouTube. So feel free to shoot any questions. I'll be fielding them to Patricia once she's done. And yeah, like first and foremost, thank you so much for your time of being here, for being part of this Global Butterfly Week, which is so far being quite a success. And anyways, I don't want to take more time from Patricia. So yeah, take it away, Patricia. Thank you.
Hello, everyone. It's
really nice to have you here and share some advances of my research here in Panama and the collaborations that I had with my peers in US and in Europe. So I would like to share some morphological and anatomical features that are really useful for the identification at the species level of these different groups of basal families that compounds the order Lepidoptera. Many of them are cryptic in colors. They have tones from brownish, grayish, and they are not so beautiful, or I can see charismatic than other species like macromoths, that they have these brilliant colors. They have this iridescence that is really beautiful and kept the sight of the naturalists and the researchers. So these groups are mainly confused by mosquitoes and other kind of things that people don't pay attention to them, but they are really interesting in their life histories and also in the high diversity of shapes and complex structures that they have developed in order to adapt to the different ecosystems that we can find in the tropics. So the next part of this talk, I will try to cover all those features and try to share a particular case, the major pest on crop of tomato, the Thornimia absoluta, and I will share some features that are really important or relevant in the determination at the species level. So let's start. So the first slide that I want to show here is a diverse of colors, shapes, and morphological patterns and different structures that we can find in the habitus of the species in the nootropic. These are pictures that I take during just one night, collecting with light attraction of the adults. You can see the different shapes, colors, different ornaments that they can have in their bodies, in their head, they can have in their abdomen, but also the different resting positions that we can find on these species or these specimens in life. So this is just a sample of the different patterns and the different resting positions that we can find on this group that make them really interesting for naturalists, for enthusiastic people, and also to research. Let's see what I have for today. We have contents. I have structured this talk in these points that we can going to see. A brief history about slide mounts, a case study based on the first slide for the identification of Lepidoptera. We're going to see the transition of the technique, how the dissection has changed through the time, and also the different, we can see the different resources, yes, that the researchers can have during the time, and they can make better dissection in order to understand other structures that in the past weren't taken in consideration. We will talk about the applications the slide mounts have on the practice of and systematic entomology. We'll also see some general terms apply on morphological studies, those based on external features and those based on internal features. I have some examples in order to contrast variability among a structure and different genera. We have some particular examples and particular species where these features have been used in order to delimitate or describe species by clarification of characters and using systematic value patterns that can be considered in order to distinguish species and also segregate what we call cryptic species complexes. So it's really important when we combine morphology and the actual techniques, the different sources that we have. So like sequences, we have different sources, not just external things or external characters. We can combine and we can make something we call integrative systematics. That is the thing or the approach that we could keep in order to advance and more have a better understanding of these groups. Then conclusions and my contact. If you would like to have any question in the future, do not hesitate and try to contact me. I will be glad to receive your questions. So let's start. So a brief history about slide mounts, a case study based on the first slides for the identification of Lepidoptera. So I have divided this line or tendons in the different continents because I have to say that each group of peers have dedicated significant time in order to understand the complex morphology of these groups. So, for example, in Europe, the first descriptions were based on dissections of the genitalia. Technical nomenclature was based on dissection of not just in Lepidoptera, but also other groups of evolved insects such as Diptera. So the first terms based on morphological features found on the genitalia were initially based on both orders, Diptera and Lepidoptera. Some relevant authors are Meyrick, Ehrlich, and Schultz. They have dedicated important time in order to contrast some features among the different families, and they describe the structure in different angles and different perspectives, giving a good understanding for their time about the structure. It's not the same technology that we have actually than the one they had in the past, but they have a good understanding of these structures by using tools like scientific illustration and diagrams. Then North America made some revisionary works in several fascicles, and they use some scientific illustration as well. And here's relevant to mention the monofascicles, some catalogs as well, were really useful in order to understand the different species. Some of the words include description, textures, original configuration, and resting position of the species in nature. Then Africa and the Middle East, they made also contribution. But I have to say, to reach all this literature is quite difficult because there are border languages. Some of the references and the journals became extinct. So it's really hard to get access to some of those publications. But in that corner of the world, they made also really relevant contributions in these basal families and in the whole order Lepidoptera. So there are descriptive works, there are inclusion of the life history records, the host plants association, and also characterization of the different inserts of the larvae. Basic dissections and scientific illustration were a primary source in order to understand the different structures found on these organisms. Some classic photographs were used as well, black and white photograph. And here's relevant to mention the work of Amsel for the Middle East, Palestine, Iran, Iraq, and part of Israel. And in contrast, for the Afrotropical fauna, the work of Janse is really important and is a really interesting reference in order to contrast with our species in the neotropical region. In Asia, in contrast, they have a really impressive contribution in all sense. They made comparative morphology as a way to explain evolutionary trends among the species. They didn't describe only sclerotides parts, but they integrate the muscular system in order to understand orientation, different adaptations of the structures, reduction, and so other patterns that can be found on the different groups. There are detailed descriptions and complex study of muscle insertion and sclerotide components of the three tagma, the head, the thorax, and the abdomen, and their appendages as well. Here, the work of Matsuda is really important and probably is one of the main references that we should consult when we are working morphology in detail, because Matsuda made a great effort in order to contrast how these structures are variable among the different orders of insects. Then the work of Okagaki, Shibatani are also relevant. Despite these researchers, something really interesting for me is that many of these researchers, they weren't entomologists as their main activity. Many of them were physicians, obstetricians, and they have other careers, but they dedicate important time in order to develop morphological studies based on or taking as role model the insects. So we're going to continue and contrast some of the first dissections based on Lepidoptera samples. Here, I want to show the first dissections made on a particular group that I dedicate some time in order to understand morphological trends within the tribe. This is a tribe Norimoskemini, and these are Povolny dissections. As you can see, Povolny made really simple dissections, just the extraction of the genitalia, the male genitalia, and keep all the structures in place without distorting the original configuration and keeping the sclerotide parts. But something curious from his dissections is that he didn't keep the membranous structures for some reason. In addition, the genitalia, the male genitalia, is completely attached. He made really basic description of this, just looking external patterns and the most, I can see, easier structures to contrast, but they are really useful if we compare them in series. In addition, the description of the female, he considered the female in his descriptions, but just basing descriptions on the sclerotized parts, such as the signum, the antrum, the apophysis, and the ostium. Then there's another trend, and we can see the use of a new procedure that can give us additional information about internal components of the male genitalia. Now is the addition of a procedure called commonly the unrolling, when we spread the male genitalia as a book, keeping internal components or internal parts completely exposed, and we can see additional patterns, we can see additional features that have some taxonomical relevance, and we can use them in order to describe, delimitate, and clarify different groups that represents a really complex fact or example in order to contrast them, because they are very close, and this procedure gives us more opportunity to see different characters, to contrast them, and add additional tools such as morphometry. So by using this step, we get additional features that in the past weren't taken in consideration. And then we see the female genitalia in another way. We keep the membranous structures and keep the original configuration of these structures in order to see additional features that in the past weren't taken in consideration. For example, the texture of the surface of the corpus bursa. It could have some relevant features that we can use them as part of contrasting different groups, complexes of species that in the past were really difficult to distinguish. In addition, we can see the ductus bursa and some relevant changes that this part of the female genitalia could have, and contrasting them between species and among tribes, and even though delimitate and clarify genera by using some of these features. In addition, we keep sclerotized components such as the apophysis, the antrum, and different structures that could be associated to these areas of the female genitalia. Then I want to show you some diagrams or scientific illustrations made by our peers in the Middle East, especially the works made by Amsel in the 1955, classic literature again, but they give us an idea of the different trends and the different features used by this author during that time, and they were valid in order to contrast the different species and describe them in a better way. So, which applications do they have on the practice of systematic entomology? Well, the elaboration of dissections and slides are really important sources for teaching, for long-term preservation of this material with different purposes, from exhibits to research, to explore different features in detail, to short-term preservation in order to diagnosticate or to determine some species that remain or could be a pest, or they could have an impact on a different scenario, not only pests, but some species could be associated with different organisms and they could be interested. And having a series or a reference collection of slide mounts can give us an idea, and we could teach the population and help giving good diagnosis by good characters or specific features that are really useful, not only external habitats. And as part of a reference collection oriented to further studies and exploration of new features, the image is just an example of one slide collection found at the Canadian Center for Research of the different group of, for example, entomological collection. See, yes, the CNC. So this is part of their collections, and these slides were part of a project related with the study of morphological features found on the tribe Norimos community, part of the world fauna of these groups. So general terms, okay, general terms applied on morphological studies. So they are variable, as we can see, and we can contrast the different references throughout the whole history from the North American peers, European peers, Middle East, and Asia. We can see that some structures may have a particular way or nomenclature, and this makes us think for now that we could create some update of the glossaries in order to keep a consistent terminology for these morphological features, because some authors can name the same structure in different ways. So we need to unify this criterion in order to get standardized these terms and delimitate, clarify these characters with better technologies using different tools that we have actually and in the past weren't possible to access. So depending on the group of insects, some structures receive a particular name, and their variations on the dissection procedures are, as I have shared before. And for example, in Lepidoptera, we can see external features, for example, development of ornaments at different regions of the body, not only on the abdomen, but we can see ornaments related with the head, with the thorax. And there are also particular modifications on the level of the wings that could be also taken in consideration by the limitation of several genera, groups, cryptic complexes of species. There are also development of specialized organs and special hairs, many of them with some relevance during the mating or the courtship of the species. And there's also internal structures that could be useful for clarification, delimitation of several genera, and solution of cryptic complexes of species. Some of them are apodiems, apophyses, and in particular, the whole genitalia as a better feature in order to study in detail. So let's see the general configuration of a moth. This is a whole body preparation that I have made, just removing the wings and keeping the body of the moth. This is more oriented to teach the different regions of the body of the moth. We can see the head is well defined with this method in particular. I have also stained what is called the thorax with their three regions, the prothorax, the mesothorax, and the methathorax. And I also made the abdomen. I keep it in the original configuration without distorting it and just sharing the three main regions of the body of the moth. The only things that I removed were the wings and part of the legs for teaching purposes. Yes. So let's go more deep. Let's start with the head. There are several variations on the head. For example, the general configuration of the head and the shape of it. Between groups, I have here two groups in particular. I have a basal family of the order. Here's a Gelechiidae, and in contrast with a macro moth. And we can see that both heads are really different between them. First, the shape, also the size of the compound eye. Some regions of the mouth, or what we call the palps, are modified, and they are really different when we contrast the different families of the order. And in particular, in the basal families of the order Lepidoptera, I can say that the palps are really important and must be taken in consideration within these groups, because they tell us different things about these families. The insertion of relevant structures. How is inserted the antenna? If it's more dorsally or frontally oriented, if it's modified, if it has some structures associated with a particular region of the antenna that the development of something we call the pecten, for example. These are some features that we should consider when we are describing species and consider them in detail. The reduction of parts and development of new ways for feeding or new strategies of feeding are related with this. For example, more basal groups remains vestigial or remain amendable, while when we go or advance along the trend of morphological adaptations of these groups, they develop pulps, they develop aproboscis, and in different ways specialize for the different tissues and the different liquids they are going to use as food. So these are some relevant things that we can associate between features and facts about the life history and adaptations of the species to the different ecosystems and habitats. So reductions of parts, development of specialized organs, they could be inside the head or they could be external. So these are things that we should keep in mind when we're describing species. For now, I just want to mention them really fast, but do not hesitate to consult me in the future if you want to know more details about any of these features and you want to see additional things or a particular way to prepare them, to see them and contrast them. Feel free to ask me in the future if you would like. Then the thorax. Let's talk about the thorax. I think in personal that the thorax is the most challenging region of the body of a moth or a butterfly. And it has a logic because probably this is the main modification of the group that give them the opportunity to migrate. They have really powerful muscles associated with the wings that give them this particular way of locomotion that they can keep for long periods, a prolonged prolongated flight with these particular modifications in the thorax. And the meso and the mesothorax are the both regions are associated with the flight and the wings. So this is probably the most relevant and complex region of the body of the butterfly of a moth. And as you can see in the picture, it have many structures. They have many sutures and what we call scrites that give them this complexity externally, but inside also the complex of the part is relevant and also have some functionality on what we call the flight, the resting position, and the courtship as well. There are several modifications on this region of the body. In addition, the development of complex structures that support series of powerful muscles. In this sense, I have to mention the furca. Is the solid region or the apophysis that insert these complex muscles and give them enough support in order to have this prolongated flight. In addition, we have something we call the tegula, and I just wanted to share how it looks. I just removed it from the mesothorax, and I wanted to show you how the tegula looks. And something particular that have this feature is that internally it has an air camera. And there are other features that wasn't taken in consideration in the past. So just by a few authors and we should keep them in consideration during descriptions. And these are the patagia. So the picture represents a sample of Erebide, but we can see that these features could be contrasted in the different families of moths, even in the basal ones. So the wings. Wings are really complex between groups. Some of them are well developed but also we can find the condition that we call brachypteris. So it's the partial reduction or complete reduction of the wing. And it's a rare condition, but we can find it within the order as a source of to contrast females and male, but also we can find this condition in both sexes. So it's really interesting, this feature, and we should consider it during our descriptions and also the limitations of some genera and in description of some particular species. And we can see in the image some of the different sprites that give articulation to the wing. As we can see, they are really small, really complex, and we can use different ways to prepare them and contrast them in shape, orientation, position, and other features that could be related with the surface of these sprites. They could have some ornaments, they could have microsculptures, and they could have different particular textures. So we should keep this in mind during our descriptions. And also, we can use this type of preparations or different types of preserved material in order to teach in a better way the relevance of these structures. I just wanted to share 3 ways to prepare wings. The first is the dry preservation of the wing, and we keep extended the wing, showing the different sprites that articulate the wing. We can also remove the whole wing and keep them in fluid in order to see the structure in detail. We can see additional hairs, specialized hairs that we can find in the wings. And also we can create teaching sources such as the one we can see at the right. And we can use this as model for teaching or during a talk to give the idea to our listeners on how complex these parts can look. So we can have this as a sample in order to teach and share more information about the different groups. So then we came to the abdomen, the third region with a high relevance during our descriptions and the limiting species. And here I wanted to make it different. I wanted to show the abdomen just being removed from the body without distorting it and without removing the covers, the scales, and the specialized hairs. So we have a ventral view of the abdomen, we have a lateral view of the abdomen, and here we can see a lateral view of the abdomen, making a particular consideration on these hairs that we can see here that are associated with a strategy of defense of that group in particular. So after a chemical treatment, I extend the abdomen as a book, I spread it, so keeping the ventral part and the dorsal part completely extend, and see the difference that it will have on the different groups and special adaptations, particularly related with the sterna. We can see these openings here that are really interesting and are associated with this structure that we can see here, that previously my peers described it as a flocculent hairs or associated with a gland as a way to defend against enemy. That particular species have that strategy, but seeing that in a morphological approach, we can see that these hairs are associated with an adaptation or a modification of the sterna, as we can see here in the image. So then we came to some particular modifications of the abdomen, and here I just wanted to show them how it looks in life, how the structure change in different family. And this is the coremata. The coremata is associated with the courtship. Yes, this is a specialized structure that has a gland and also specialized hairs that helps to diffuminate the pheromone among the species. And they have different structures to make it possible. So as we can see in the same family, Erebidae, the structure can look very different, but also is functional during the courtship. And we can find it also in groups like the Pyraloidea, and in basal families of the Orden Lepidoptera, such as the family Gelechiidae. And if we see in detail, this structure could be inserted in different regions of the abdomen, not just related with the subgenital plate or the last abdominal tergites. No, we can find it more related with the basal area of the abdomen. We can see it inserted laterally, dorsally, ventrally, or a combination of this. So as you can see, it's really complex, but we have a lot of different features and really interesting facts about just a particular character and how it varies through the different families or groups of moths in this case. But in butterflies, some of these features, we can see it also, but we need to contrast. That is why we need to unify criterion. We need to talk more between lepidopterists, because I think these things deserve to take them in consideration and have these deep discussions about characters and limitations of them and see what our previous peers didn't solve. So this is really important to consider. So then we came to the genitalia, the males. In this particular case, I just want to share just one example, the species Cadra cautela, and it's a pyraloid. And I just wanted to show the different features or the different characters that we can find associated with the male genitalia and how it varies through different groups. I have this particular example that we can see. I will take just one character from this image and we are going to contrast them that with the different families that I have in the next slide. So I would like to contrast here, for example, the shape of this structure in total called the valva, and the cuculus is this external margin. So let's take a particular care to that region of the male genitalia, and we're going to see how it varies through different species and different groups. So as you can see, I keep the original configuration of this species in particular and just give particular detail to the different structures related with the genitalia, the vulva, the saccus, the vincular area, the natos, and the uncus. These are structures that are really important during descriptions because they give us the enough solid characters in order to contrast the species. They are really interesting how they vary, how they change through the different families and the different groups. And also the corpus phallus. Here, when I detach the corpus phallus, I keep the membranous areas as well. Some authors, they remove them, but yeah, I think it's important to keep the area associated with the basal part of the corpus phallus, that area called the cecum, the lamina ducti ejaculatory that could be associated with that area as well. So then we came to another species. This is another male. This is the particular case of the tomato. No, this is the potato tuber moth. Yes, this is called the Thesia solanivora. As you can see, this moth is completely cryptic. It's brownish, and many species from the genus Thesia, they looks quite the same, brownish and without any external features that give us the enough sense to say this is that species and we can segregate it from the rest. No, we need to dissect in these groups. So I just removed the genitalia by using the technique I have mentioned before, the unrolling. I spread the whole male genitalia in like a book and keep the external features exposed, internal features exposed, and also the corpus phallus and the areas that have some exploratorization related with them as well, with that as well. So I have pictures of the habitus of this moth and the genitalia, and we are going to see the feature I have mentioned before, how change the vulva. So here we have, in contrast with this, here we see something more, we can say like it's more circular at the distal part than here, that is more elongated and with some curvature inwards. Yeah. So here's the cuculus, and we have the whole valva. As you can see, the shape, the configuration, and the orientation of the valva give us more information, and it's really different between both species, if we contrast them. In addition, the other structures that I have mentioned before. And here is what I mentioned about the corpus phallus. If we keep the basal area of the corpus phallus, we can see that it changed in a drastic way in the species. We keep a cycle more prolongated basally, and we see an sclerotide lamina here, which is called the lamina ductis ejaculatory, that some authors previously didn't consider it. They just removed the sclerotide part, and they just don't consider that part, and I think it deserves to be considered on descriptions. Then the genitalia, the females. The females weren't considered in the past for some reasons, but now we can say that the female genitalia have some relevant features that need to be taken in consideration during our descriptive task. For example, the shape of the corpus bursa, we can see a drastic change in morphology or in shape within this species and this. Both are from the same family, but are different species, completely different genera. We have here the case of Absoluta, and here in contrast we have the case of Aristotelia. Is another genera or genus of the family Gelechiidae. And we can see that it's really variable, the shape of the corpus bursa in this species. Also the shape of the signum. We can see here a hook-like signum, but here it looks more like a patch with serrate margins here. There are also drastic variations on the ductus bursa. Here is very short, but here is really long and with some helix or curvatures among it. And we also can see differences in the papilla and also in the length of the apophysis, for example. So the case of Torimaea absoluta, this is a species that is well known in the world by its major impact on the tomato crop. The larva in particular, the different inserts of the larva could cause several damages, serious damages on the production of tomato and the different stages of the crop. So it's relevant to know well this pest in order to take a good decision in order to control it in the best time possible. Because if we let the pest take part of our crop, at the end, we are not going to get the results we expect. So it's relevant to all the people related with farms and crops, and in particular with the tomato, know well this pest and know to distinguish it very well from closer species. So this is another case of critic species without external features that can be used to distinguish. In this case, in particular, we need to prepare dissections. We need to prepare these procedures in order to give a robust diagnosis of the species and take the better decision in order to control it. So here we see the male and the female of Absoluta. This is part of a work that I did in collaboration with my peers from the Smithsonian. And here we use several structures or several features in order to delimitate and give a clear diagnosis of the species, considering the morphology as a target point in order to delimitate and also give a better position based on morphological feature of the species. So here we consider the different structures, and just keeping the example that I gave before, we can see the shape of the valva here is really different. It's more reduced. Also, it has some ornaments on the surface of the cucular area. But also we can see structures like the vinculum, the development of these areas with emargination. We can see also the sac is prolongated, the saccus. Also, the shape of the corpus phallus is straight, completely straight. But also we see the sacum, how it changed. It has more curvature at the basal area. It has some punctures on the surface, but also the area with membranous parts are kept. In addition, I can say that the uncus here is relevant in the diagnosis of the species and other features like the three escarites that compounds the natos. In addition, the female, here we can see the corpus bursa extended, the signum, also the spermatica is another features that need to be considered. And I think that it will give better description and is contrastable between species and between genera as well. And the apophysis, the length of the apophysis, the level of sclerotization, and in some of these structures tend to be melanized. These are some features that need to be considered in further descriptions as well. And well, I just wanted to share characters because many people who study these groups, they prefer to stay just by pictures, seeing habitus, but these are really complex groups that they need something more. They make us to see a little bit more in detail and also use the different methods, the different tools that we have actually in order to contrast them and describe them in a better way. So this is a source that we must consider, especially in cryptic groups that are many in our tropical countries, in the neotropical region. We have a lot of shapes, we have a lot of particular adaptations to the different ecosystems. So as these adaptations happens, we can see that represented on the morpho-anatomical patterns found on the species. So that's why we need to see a little bit more. We need to get further, and we need to study these structures in detail. And using different methods of preservation as well, not just keep the slide mount and that's it. No, we can keep the same species, but in different ways to prepare it and see different angles of the same structure, different shapes, textures, orientation, insertion, interactions with other systems, and that gives us a better understanding of the group. So that's what I wanted to share for now. And just keep it in mind and think a little bit more when you see a moth that high complexity that could be found on the body of these organisms. So conclusions. The study of the anatomy and morphological features on different orders of insects has been an important fact to inspire and innovate in a wide variety of disciplines. As we can see, have seen on the first part of this presentation, some examples are variations of the dissecting procedure through how the whole history of descriptions, we can say, and different criterion on taxonomical descriptions. The comparative study of morphological features on the Orden Lepidoptera has been a key point for studying evolutive relationships among groups, the limitation of relevant regions and structures, and the innovation of the dissecting procedure has revealed new structures with a high relevance in systematic studies. So next time when you prepare any dissection, particularly related with the last abdominal target and involving the genitalia, try a little bit more. Go to the unrolling and try to visualize membranous structures related with these internal parts and how they are connected, and possible interaction with muscles. This could be really useful in a functional morphological approach, but also we can have different features in order to describe better a species. That's the lesson taken from our peers from different corners of the world. Thanks. Thanks in advance and hope you have enjoyed this talk. And if you have any question, do not hesitate and feel free to send an email or just write me through the different sources, such as Instagram or whatever. Thank you very much.
Thank you so much. Thank you so much. Like, to be honest, after all the hiccups that we had in setting up this talk at the beginning, I'm glad that everything went great, and to be honest, was worth it. I learned a lot. wow, like there's a reason why I admire taxonomists and why I leave that work to you. But it's a really really important work, and that and I'm sure there's going to be a lot of people that are going to be interested in this once they watch the video. And of course, I'm talking in your name, but I'm sure that you'll be happy to maybe reply to some emails or questions if they send them your way, right?
Okay.
So anyways, we only have like 3 minutes for for Q&A, but I'm not sure if we have people that have any questions or not. Let's just check the chat really quick. Let me see. Streaming. No, and here. No as well. So I mean, we did have at some point a good amount of people watching. I'm gonna share the link with you afterwards, Patricia, if you wanna share it with your networks and whatnot. Feel free to do it. But anyways, I really wanted to thank you for your time and like sharing all this amazing knowledge that you have inside your brain, which must be like this size. So congratulations. And yeah, so we need to hop on to the next talk that is starting just in 5 minutes. Which comes all the way from Argentina. So feel free to hang around, Patricia, well, on the other side of the screen, just to watch this next talk. But anyways, again, thank you so much for all people watching. Thank you so much for being here, supporting Global Butterfly Week. And yeah, let's just keep pushing conservation.
Thank you very much for your attention. Thank you so much. And do not hesitate to consult me and contact me through the different networks and different sources. I let my email, so if you are interested to knowing more about the different morphoanatomical features and how they can be used on different ways in order to teach, in order to know more about the different adaptations of these groups, feel free to contact me. I'll be glad to share what I have through all these years, and we could have some interactions, and that will be benefit and could be a good integration of the different criterion. And at the end, we can give some structure to the different terms and do not have this different terms to the same structure. We can unify criterion. So this is important, and stay tuned. Bye.
Awesome. Thank you, Patricia. Take care. Thanks, everyone.
Bye. 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 Bye.